Hello AP Biology students, we're going to go over the 10th section in Unit 1. And in this section, we're going to talk about my favorite type of macromolecule. It's known as proteins. The monomer of a protein is known as an amino acid, and the polymer that it makes is called a polypeptide. The elements that we find in proteins are carbon, hydrogen, oxygen, and nitrogen. So one defining quality of protein is that it contains nitrogen. Remember our first two macromolecules of lipids and carbohydrates. They do not contain nitrogen. Proteins do. The type of bond that forms between the monomers is known as a peptide bond. And basically, these proteins act as workers of the cell. I love this little graphic here. You can see the amino acids bonding together to form a polypeptide. And then that polypeptide is going to have a specific shape that determines its function. And that function is going to help the cell complete some task. Again, think of these proteins as being the workers of the cell. One of the reasons why proteins are my favorite macromolecule is that they make up more than 50% of the dry mass of a cell. So most of your cells are proteins. And there are hundreds of thousands of different types of proteins, and there are new proteins still being identified. Now, something that's really important about this whole unit is this last bullet point here. The sequence of amino acids is going to be important because it's going to determine the shape of the protein. And my AP Biology students are going to hear this 1,000 times. The shape of something determines the function of that thing. In other words, structure determines function. So in this case, the amino acids and their sequence within the protein are going to determine the structure and then function of that protein. This is an example of a polypeptide where we can see long chains of amino acids binding together, and the sequence of those are going to determine the structure of that protein. Let's talk about amino acids for a little bit first. There are 20 different types of amino acids, and they have some common characteristics or common functional groups that we're going to see across all 20 amino acids. In all amino acids, we're going to start out with a central carbon atom here that's going to be attached to this amino group, which is NH2. This carbon is also going to be attached to a carboxylic acid group, which is a COOH group. Lastly, this carbon is also going to be attached to a hydrogen. These are the common functional groups that we see between all amino acids. Now, the one different part of an amino acid is the R group. This is the variable chain group. And if you remember, this R group is going to have a host of different types of atoms that could be contained in this one little group. I know it's kind of hard to see, but these are the amino acids. You can see the top portion is identical between each amino acid. Below each central carbon is the variable group, which is different between each type of amino acid. So again, there are 20 different amino acids. Each of the amino acids have functional groups that are exactly the same between the amino acids. What is different between the amino acids is this R group, and the atoms that are in that R group are going to determine what type of amino acid it is. You could have acidic, basic, polar, and nonpolar amino acids, and they all are determined by the atoms within their variable group. Now, like I said before, the structure of a protein is going to determine its function. To determine the structure of a protein, we have 4 different levels of folding. We have the primary, secondary, tertiary, and quaternary structures. I'm going to briefly talk about each of the different structures here and then show you a graphic that helps illustrate this concept. The primary structure is just the linear sequence of amino acids, and these amino acids are going to be joined together through covalent bonds, or otherwise known as peptide bonds. The secondary structure is when the polypeptides or the amino acids are going to coil into things called alpha helixes and beta pleated sheets. This coiling is going to be due to hydrogen bonds between adjacent or close by amino acids. The secondary structure is going to be very noticeable or identifiable by the alpha helixes and beta pleated sheets. The tertiary structure is when these alpha helixes and beta sheets are going to form a 3D globular protein. This is because these alpha helixes and beta sheets are going to start folding more on each other. And these again are going to be due to the characteristics of the amino acids. Now, not all proteins are going to have this last structure, which is known as a quaternary structure, but some are going to have several polypeptides forming a larger protein. And again, these polypeptides are going to join together based on the amino acids that make them up. So again, some proteins are going to stop at the tertiary level, while other proteins are going to stop at the quaternary level. Here's an example of the 4 different levels of protein structure. You can see in the first, or the primary structure, we just have the sequence of amino acids. The secondary structure is made of alpha helixes and beta pleated sheets. The tertiary structure is when these alpha helixes and beta sheets are going to bond together, forming a globular structure. Now, some proteins are going to have this quaternary structure where there are several polypeptides that join together. The structure that the quaternary and the tertiary structure are going to form are going to be based on the sequence of amino acids found in the primary structure. So again, the primary sequence, or the linear sequence of amino acids, is going to determine the shape and function of the protein. I like to show this level of secondary structure where you have the beta pleated sheets here, and they're going to form because of hydrogen bonds between the amino acids. Alpha helixes are also formed by these hydrogen bonds, but they have this corkscrew-like fashion, or corkscrew-like structure. One last thing I like to mention about these proteins is something called denaturing. And denaturing is when the proteins unfold because of heat or pH changes. Basically what happens is the protein will unfold because the hydrogen bonds between the secondary and tertiary structures will be broken because of these changes. You can see this normal protein unfolding because there is some change in heat or pH. Now you can see these proteins are going to lose their structure, which is also going to mean they're going to lose their function when denaturing. This is an example of a polypeptide folding, making a protein. Now this polypeptide is made of amino acids. These amino acids are represented by the little structures coming off of the linear line. In this video, you can see that the linear sequence of amino acids are going to determine how the protein folds. And that protein then is going to have a specific shape, which is going to determine its function. And these functions are going to help the cell complete different tasks. I like to show a bunch of different examples of proteins like these to show students that anytime you see these corkscrew-like alpha helixes or these beta pleated sheets, which are represented by arrows, you are looking at a protein. All right, here's another good meme. What part of the amino acid does a pirate like the most? The R group. Get it? Because it's R like the remain. Okay, bad joke, I'm sorry.